Hi everyone, I want to use this video to give us the basics about cell and tissue culture. And so after this video, hopefully you'll have a pretty good idea of the difference between primary cells and cell lines, about what it means to feed the cells, to passage the cells, all the requirements for growing and maintaining cells in culture, and then also a little bit about chamber size and cell counting. All of this is going to be what we're really introducing and getting you used to this week in lab. So hopefully after this you'll have a bit better understanding, but definitely bring questions, and we'll still talk a little bit about this as we introduce the lab, but this will allow us to move a little bit faster right into the actual lab work on Wednesday. So my special thanks to Brianna Jones, the TA, who really put together a lot of these slides and helped me get this going. But let's talk a little bit about tissue culture. So first of all, primary cells versus cell lines. So primary cells are cells that are derived directly from the source. So what do I mean by that? Well, that means if you're studying kidney cells, for example, this would be taken directly from a biopsy or a tissue sample from the kidney, and then you would start to grow those cells, and you would have kidney cells in culture. That's a primary cell. It's direct from the source. There are also other types that you've probably heard about, embryonic stem cells. These are still primary cells because they're directly from the source of an embryo. Induced pluripotent stem cells, this is where you take a differentiated cell from the body. Usually it's a skin biopsy or white blood cells, but you actually add some factors to de-differentiate them. So it takes these differentiated cells, de-differentiates them to embryonic stem cell-like cells, and then you can re-differentiate them into various types of things. And then there's also adult stem cells of bone marrow cells. We'll actually be using bone marrow cells, hematopoietic stem cells from the bone marrow, later on at the end of the semester. But these are all examples of primary cells. So remember that they're from the source itself. The downside to these is they can be difficult to get and to isolate, and they also can be difficult to grow because they're very specific to that region where you got them from. And they're also not able to grow indefinitely because most cells in your body have a limited lifespan, limited number of divisions before they start to die off. That's why we usually don't have cancer under normal conditions. So they can be a little bit more difficult to work with, but the benefit obviously is that they are the most representative cells of whatever you're studying. If you want to study the kidney, getting primary cells from the kidney is the best way to go. Cell lines then are immortalized versions of cells. So they might have started off as primary cells, but they are now immortalized, and so they can grow indefinitely. So before I get into how this happens, the obvious plus side to this is that they are much easier to grow, and you can grow them indefinitely. The downside is because they're immortalized, and as they continue to grow division after division after division, this generally makes them a little bit further and further away as the longer they've been alive and the longer they've been used from their original source. So if you have a kidney cell line, it's not going to be quite as representative of kidney cells as a primary cell from the kidney would be. However, they're going to be a lot easier to grow, a lot less costly, and you can continue to study the same cells over and over again. So how do you immortalize them? Well, some are naturally immortalized. So if you derive your cells from a tumor source, then the tumor is naturally immortalized. That's one of the reasons it's made to be a tumor. So there are some tumor-derived immortalized cell lines. You can grow them, and then over time, your primary cells, a few of them might have spontaneous mutations that make them more tumor-like. So these would be spontaneously immortalized cells, and then as you keep growing, the rest will die off, and you kind of start to maintain only the ones that have become immortalized. You can actually force the cells to express an oncogene. So this oncogene basically tells them to continue to divide, just like it would in cancer. And so if you transfect these cells with the oncogene, then you're basically causing these cells to continue to divide. And these are often transfected using viral transfection agencies. And you can activate telomerase. So this basically is just reactivating that telomerase that re-extends the telomeres so that the cells don't die after a certain number of divisions. And one of the other big benefits to cell lines is that you can actually start to transfect these cells with other types of genes. So if you wanted to force the cell to express a gene that it doesn't normally express to really study what that gene does to the cells, then you don't really want to have to re-transfect them over and over again. You want a cell line. A good example of this is, for us, we actually are studying HPV. So we're injecting an HPV oncogene into the cells and really seeing what other cell cycle machinery that affects. Why does it cause cancer? But when we try and just transiently transfect, just kind of add it to the cells, only about 10% of the cells actually get the gene inside it. And so if we look at those cells as a whole, we're actually studying 90% of the cells that aren't expressing that HPV gene that we want them to. So if we inject the gene we're interested in, and then we start to select them, now we're selecting for the ones that are expressing that, then we can get an immortalized cell line of these cells that are now expressing the gene that we've added on. So you have your plasmid with your gene of interest. In our case, it's an oncogene from HPV. We use some virus to basically try and get that gene plasmid into the cells. Not all of them express that, but a few do. So we select for the ones, so these would have an antibiotic resistance or something that allows you to select for it. The antibiotic kills all the cells that don't have it, and then we would be left with the ones that do have that transgene. And if it's immortalized and you continue to grow it forever, now we have a cell line where all the cells have that gene that we want. All right, and so I think I've talked a lot about the pros and cons of each already, but the primary cells, directly from the source, most representative. Immortalized cells, much easier to grow. Another advantage of the immortalized cells is that you're studying a clone, so it's going to be the same now and in other trials and so forth, whereas primary cells, you have to get them, basically refresh them, get new donors each time. So the advantage there is that you have a more representation of a full population. Here you have a representation of the one source that it originally came from, but you also have more variability with these primary cells, less variability with the clone. So what does cell culture look like? Well, we're trying to grow these cells in these various flasks, and there's a lot of different types of flasks you can grow them in. We'll use some multi-well plates sometimes. People will use 6-well plates, large round dishes. The ones we're going to use for the most part to maintain our cells are these T75 flasks. So these are dishes where the cells are going to grow on the bottom, and you'll have the media on the top, and we have basically a filtered air passage at the cap so that there's some gas exchange going on. These cells will grow in a tissue culture incubator. So these are CO2 incubators. This will keep the temperature at 37 degrees Celsius, which is equivalent to approximately 98.6 Fahrenheit. So that's body temperature. And these also will maintain 5% CO2. So 5% CO2 seems high if you realize that basically the outside air is much less than 1%, usually around 0.2% or even less than that. But what happens is the cells in your body, because they are undergoing cellular respiration, they're breaking down glucose and producing CO2. And then that CO2 goes from your cells into your bloodstream, onto your lungs, and then diffuses out into the air. That's why we're breathing out CO2. So the actual concentration of CO2 at your cells is about 5%. So by keeping these in a 5% CO2 incubator at 37 degrees Celsius, we are mimicking the natural environment that cell, the mammalian cell at least, would be growing inside your body. Incidentally, this is one of the reasons that if you've ever watched Apollo 13, the reason that 6% CO2 is so important. If the chamber of their spaceship got up to 6% CO2, they were going to die, because that reverses the diffusion. If your cells are at 5% CO2, that CO2 is higher than the CO2 in your blood, so it diffuses to the blood. In the blood, it's higher than the lungs, and in the lungs, it's higher than the outside air. But if the outside air becomes 6%, you're actually no longer able to get rid of your CO2. If anything, the diffusion is coming from the air on into your cells. And that's why they would have died if the chamber made it up between 5 and 6% CO2. We also need to provide these cells with nutrients. So there's media. These are buffered medias. And these media, there's a lot of different variations, but most of them are going to have a lot of essential amino acids, a lot of vitamins, and definitely some glucose, some energy source. And then we usually add 10% fetal bovine serum. So in the serum, there's a lot of growth factors and things like that your blood would normally be giving to the cells. And then we add antibiotic and antimycotic. This is just a way to really hedge our bets against bacterial or fungal infection. Although even with that antibiotic and antimycotic, we still, as I'm going to tell you, have to be extremely careful to make sure we maintain sterile conditions and don't let those things get into our cultures. We also have two different types of cells. We have suspension cells. These are the ones that don't really attach to the flask, so they might live in suspension in your body. And then we have adherent cells that activate cell-cell adhesion molecules and also cell integrin or cell molecules that will allow them to attach to the plastic flask. So these are going to adhere to the bottom of your cell culture dish. These are going to float in the media. All right, so if all these cells need those nutrients that we saw in the media, then that means they're also going to be using up those nutrients. By the fact that they're alive, they're going to be using up those essential vitamins, they're going to be using up the glucose, they're going to be using up this growth factors and so forth. So you need to feed the cells, basically providing them fresh media with fresh nutrients. So how do we do this? Usually this has to happen every couple of days, although as I'll talk about, we really will only feed the cells every 4 days because in between those days, we're also going to be passaging the cells, and that gives them fresh media automatically as well. But how do we do this? Well, if the cells are adherent, it's really easy. So they're stuck to the bottom. So if you take a look at your cells, you see that they're stuck to the bottom. All you have to do is remove 3/4 of the media from the flask and give them fresh media with fresh nutrients and so forth. Why do we leave 1/4 of the media in there? Well, these cells actually produce their own growth factors and a lot of survival factors that help each other grow. So it actually is beneficial to leave some of the media that's accumulated the factors that these cells are making themselves, but we certainly want to give them more nutrients along the way. Suspension cells are a little bit harder because they're in the media. You can't just remove 3/4 of the media. If you do, you're going to remove 3/4 of your cells. All right, so what you have to do is remove the media. The cells are in that media. You're going to spin down the cells in a centrifuge. That makes the cells, you have a little pellet at the bottom, and you don't want to spin them too fast. We're just spinning them gently. We want them to stay alive. But now that the cells are at the bottom, now we can remove 75% of the media, replace it with fresh media, resuspend the cells, put them back in the culture, and they're growing again. So that's feeding cells. Obviously, as cells are growing, they're going to get more and more dense because these cells are dividing. That's what we want them to do. So we call this confluency. Confluency is a measure of how densely packed those cells are in our culture. So this would be an example of 10% confluency. Only about 10% of the bottom of a flask has cells on it. Then a little more confluency, 30%, half confluency, and then you get up to a high level of confluency where these cells are running out of space to grow. And before they get to this point, that's when we need to give them more room to grow. We do not want the cells to reach 100% confluency, because if that's the case, they are now stopping each other from growing, and you're now going to select for the cells that are even more tumorigenic, because any cell that's still able to grow are ones that have to grow on top of the other ones. And that's going to cause more mutations, that's going to change our cells. So we want to make sure that we are giving our cells more room to grow before we get to that 100%. Usually somewhere in the 78 to 80% we want to split our cells. So how do we do that? When that happens, we need to do what's called passaging or splitting. Those are two fairly synonymous terms to give them more room to grow. All right, so if the cells are in suspension, now this is actually easier because the cells are in suspension. Now we're trying to get rid of a certain percentage of the cells. So we can basically, if we take out 80% of the media, if we have 10 mils of media, we take out 8 mils, we've just taken out 80% of the cells, leaving only 20% left. If we give them fresh media, we've just split those cells 1 to 5, because now the cells left in our flask are only 1/5 of the cells that were there before. And with the fresh media, they have plenty of room to grow. They can grow up until we get high density again, and then we'll split them again, and we'll keep them growing with room to grow as well. Splitting cells for adherent cells is a little more difficult, and this is what we're doing with our cells, actually. You're going to have your cells in a culture dish. All right, they're growing. They are adhered, and so in order to split them, we actually have to de-adhere them. So we're going to use an enzyme called trypsin that also has EDTA. So what EDTA does is it's a divalent cation chelator. It removes the magnesium, the calcium, all these divalent cations that your adhesion molecules, cell-cell adhesion, cell plastic adhesion molecules need to function. So with EDTA in the media, that kind of causes them to become unfolded and floppy and non-functional. And then trypsin actually even goes a step further and will cleave off all the extracellular domain of our adhesion molecules. So the trypsin EDTA will release our cells from the flask, and now we can do our split. So if we have our cells released from the flask in 10 mils of media, we can take 9 mils of that off, collect those cells. We've just collected 90% of our cells. If we add 9 mils of fresh media, no cells in the fresh media, back in, now we have just split our cells 1 to 10. We've left 10% of the cells in the flask, and now they have room to grow again. All right, so that's the idea of splitting. You're basically removing a certain percentage of your cells so that you've diluted the cells that are remaining in the flask. They will still have room to grow. Whenever you're doing this, you want to add another passage number, because the more often you do this, you have the longer these cells have been alive, and the more chance there are for mutations, and the better chance there is for the cells to have changed. So every time you do a split or a passage of the cells, you're going to add a passage number, because we want to keep track of how old the cells in our flask might be. All right, so this is just that kind of idea again. So here's a confluent flask of cells. Right, so we are going to add trypsin. We're going to make those cells dissociate from the bottom so we can get them. After subculturing, we're going to put them back down. We've removed a lot of the cells, and then the remaining cells have some room to grow again. Right? Now, the cells we removed, you can do a number of things with them. You can freeze them down, cryopreserve them, so that later on, if we need cells again, we can go back and we have a low passage cell. So let's say the ones that we kept growing end up with much higher passage later on. Well, if we froze these cells down to low passage, we can go get them again and start that cell culture over again with the same types of cells. We can also collect them to do experiments on, and we're going to do a lot of these experiments. We're going to stain the cells. We are going to test the cells for chemotherapy agents, what effect those have on the viability of cells. We're doing a lot of these things with our cells over the course of the semester. We'll talk more about that as those events come along. All right, so one of the things we are going to do though this week, because you have to do this over the week in preparation for next week, is to create cell chamber slides. Right? So when we're growing cells in our flasks here, we actually can't use this on a fluorescent microscope or a bright light microscope, only a phase contrast scope that lets us tell if the cells are alive or not. In order to really study those cells by staining them or using fluorescence microscopy, all those things we might want to do with them later on, we have to get them onto a chamber slide. So basically, this chamber slide is almost like a cell culture chamber, but on a glass slide. And so when we collect our cells after a passage, so when we collected our 90% of the cells that we took out so that these cells have room to grow, we can take a small percentage of those cells and we can put them on a chamber slide. And they're going to grow on the chamber slide. If they're adherent, they'll adhere to the bottom. And then at the end of that, we can use the slide to stain them. We can get rid of this plastic top, and we'll do this again next week. And basically, in doing so, we now have a slide with our cells grown on them that we can image, stain, do whatever we want with them. All right? So that's one thing that you are going to do, and we'll talk a little bit more about that this week. Another thing you need to know how to do that we're going to practice this week is be able to count cells. So we sometimes want to know exactly what concentration of cells we have so we can make sure that we're putting the right number of cells in a certain plate or in wells of a dish and so forth. So how do we count cells? Well, to do that, we use what's called a hemocytometer. Cyto basically means cells, and so we're basically counting cells with a hemocytometer. And on this hemocytometer, I know this is a little bit fuzzy here, but there is this really microscopic grid that you can see under the microscope. Within this microscopic grid, there's basically 9 squares, and each of those 9 squares is actually subdivided as well, but this would be 1 square, a second, third, shown here in black, fourth in the middle, 5, 6, 7, 8, and 9. So it's kind of like a tic-tac-toe board with many demarcations in between. The significance of this is within any one of these kind of middle-sized squares, any one of these 9, that is set up perfectly, so with a cover slip on the cells, and the cells in that area, that is exactly 1/10⁴ of a mil. So if you count the number of cells in one of those squares, you know how many cells are in 1/10⁴ of a milliliter. All right, so 0.0001 milliliters, that's how many cells are there. And if you're able to count how many cells are in 1/10⁴, you can extrapolate how many cells would be in a mil. So, for example, if I counted 50 cells in here, and I diluted my cells to get this by 10, I know that my actual concentration is tenfold more. And sometimes you have to dilute because you don't want to be counting to thousands of cells. So if I had counted 50 and I had a dilution factor of 10, well, that's 50 times 10, 500. And this is 1/10 to the fourth of a mil, so I multiply it by that, and that's how many cells per mil I would have. So it allows you to basically count a very small volume of cells and extrapolate to the much larger volume. And we'll use a hemocytometer, but this is basically what this will look like. You can see the cells on our grid here, and we're going to count these cells, and you're going to learn how to know your concentration, because we're going to use it later on for things like creating standard curves, so different dilutions of cells along the way. So this is just an assay that kind of reacts with live cells, and the more cells you have, the darker the reaction, so you can kind of see a cell curve going. And with this, we'll be able to determine if our methotrexate, our chemotherapy agent, kills the cells and at what dose is needed to kill and things like that. But that's coming up later on, that cell viability assay. But that's why we need to know how to count cells. So that's what we're doing this week. We're going to learn and perform tissue culture, because you're going to maintain cells over the course of the next 6 weeks that we're going to use. And we're going to learn how to make our chamber slides and how to count cells so that we're prepared for some of those other things that we're going to do the rest of the semester. So just real quick, what cells are we using? We are using L cells. These are fibroblast cells, which are cells that kind of help make your extracellular matrix, your connective tissue. These are cells from mice, fibroblasts. All right, cells in culture. We need to be really safe. This is biohazard safety level 1, which is the lowest level because these cells aren't very dangerous, but we're still working with mammalian cells. And we need to be cautious for our own safety, and we also need to be cautious so that we don't contaminate the cells. All right, so there's no eating, drinking, nothing in the laboratory with regards to your mouth chewing anything. Anytime that you work with cells, all of your pipettes and everything should be bleached, but do not wipe the TC hoods down with bleach. That will cause them to rust. If you do spill any media and so forth, we're actually going to clean that up with 70% ethanol. So 70% ethanol, basically 70% ethanol, 30% water. The ethanol basically permeabilizes bacterial membranes or any cell membrane, and then the water rushes in by osmosis, and this basically causes the cell to die. This is actually one of the reasons why hand sanitizer works. Hand sanitizer is just basically 70% ethanol killing bacteria and viruses, etc. on your hands. And the hand sanitizer is just jazzed up with a little bit of lotion and something to make it smell better. So kind of the same idea. So basically we're always going to sterilize the hood with 70% ethanol. Another thing is don't open any solution. Media is very good for things to grow in, so if you open it up out in the air, bacteria are going to grow in it. So we're only opening those things in our TC hoods and so forth. Keep solutions closed as often as possible, and again, don't open them anywhere except the hood. And basically, know what you've touched. If you've used a pipette to suck up your cells, you don't want to go back into your stock of solution, because if you do, even if the pipette's empty, there's cells stuck to it. And as soon as you put anything that's touched cells into your media or your stock solution, well, that now has cells, and you're going to be able to cross-contaminate anything else you use. So once you've pulled up cells, do not go into your stock solution with that same tip. All right? Remember, it only takes one bacteria cell, one fungal spore, or even one mammalian cell in your stock solution to really start to grow, contaminate, and can kill your whole culture. So we need to be really careful with this, and we'll stress this more in the class. All right. Look forward to talking with you some more. Last two things, and we will talk about this more as well, but we're going to work with an e-notebook. And so one thing is you're going to keep track of what you're doing and what you have done. We have to feed the cells every 2 days, and usually they will need to be split every 4 days. By 4 days, they're going to be nearly confluent, and you'll have to split them. All right? So basically, unfortunately, that means over the next course of the next 6 weeks, you and your partners are going to have to take turns and come in and either feed the cells or split the cells, depending on which days it is. All right? But that will really get you some great experience with tissue culture, and we will use these cells. So there might be some weeks where you have to do some extra things over the course of the week in preparation for the lab. Like this week, you're going to make your chamber slides. All right, and then finally, the e-notebook. So the big project for this is maintaining a laboratory notebook And I say e-notebook because this can be on a Google Drive, Google Documents, Google Excel file, and so forth. But basically, this is going to encompass everything that you've done in detail, and something that you could go back to or hand to a lab mate, and they would know exactly what was done, what you saw, and so forth. Right? So for the tissue culture project, there's a few things you need to do. We're going to expect images of cells at low, mid, and high confluency. So when you come in to feed the cells at mid confluency, take a picture. Right before you split them one of the times, take a picture. Right after you split them, take a picture. And you'll have those. This is because then you know what healthy cells look like at all those confluencies. We also are going to expect you to keep a log of what was done. So whenever you come in to feed the cells, just log it down. If you come to split the cells, log it down. So what was done, the date, who did it, did the cells look healthy? Get used to always looking at your cells on the scope before you do anything to them and right after you do anything to them. Know if and when they're alive and how confluent they are. If you split the cells, what was your ratio? Did you split them 1 to 10, or are they growing really fast and you had to split them 1 to 20 and so forth? And if there's any extra notes you had to do, that should all be in there for the tissue culture. The procedures using the cells, for example, our staining procedures, our cell viability assay, Western blots, things like that. Every time we kind of do a procedure using the cells, you should have a purpose. So what are we doing and what is the purpose of this? All right? Immunocytochemistry, what are we staining for and what do we expect to see? This doesn't have to be long, but you want 3 or 4 sentences on what you were doing and why you were doing it. Because if you came back to this lab notebook 6 months later, you need to remember, oh, what was I doing? Oh yeah, okay, now I remember what I was doing and why I was doing it. Get used to kind of having that purpose. You don't have to write the protocol. You'll get a protocol from us, but you do need to have a link to the protocol. And this protocol and procedure can be copied from what we give you, but it then needs to have your own hints about what was done. So as you go along, if there's something that you had to add to kind of really understand it, make sure you add it so that if you came back again 6 months later, you would know what was done. Information on things to consider, changes you made to the protocol, your final images, graphs, or results. So when we stain something, images from the slides. If we have graphs from your viability assay, kind of all the results of that, and then a brief conclusion in the next step. So this is not a lab report or anything, but it's something that is extensive enough that you would know what you did, what you saw, what the conclusion was, why you did it, etc. If you came back and looked at a good lab notebook several years later. All right, this is a much longer video than I usually would like to have, but I think this really sets us up for a great semester and allows us to get started pretty quickly on Wednesday. Please bring your questions. We will talk more about all of these things and expand on it a little bit, but this will give us a chance to really just focus on the lab itself on Wednesday. All right, thank you, and I'm looking forward to this, everyone. Bye.